Hello, my name is Anne O'Donnell Luria, and I am a clinical geneticist and researcher at the Broad Institute of MIT and Harvard and the Boston Children's Hospital. Today, I have the pleasure of telling you about the use of the Genome Aggregation Database, also known as gnomAD. In every genome, there are many rare, potentially functional variants. However, taking a step back, every genome has about 2 million or more high-quality variants in a variant call file, or VCF. About 20,000 variants within the coding or exome regions and the edges of the exons. About 500 rare missense variants, and a third of which are predicted damaging by in silico predictors. So even as we add tools, we still have a lot of variants to consider. If we say, let's only look at loss of function variants, we all have about 100 of those, including about 20 that are homozygous and 20 that are rare. We have about 100 rare variants in known disease genes, about 50 reported disease-causing mutations, although I will say this number is a slightly old number and it's been falling as these databases get better. And we all have 1 to 2 de novo coding mutations. These are a place we look for variants that could potentially be contributing disease, but we all have these. And on top of this all, there's an unknown number of sequencing errors. So as we think about Mendelian disease analysis, which there are many different types of reasons to do genomics, but Mendelian disease analysis is one. One of the questions we encounter is how can we identify the pathogenic genetic variants in the sea of benign variation? Well, with the power of 7.7 billion people, we know that based on given mutation rates, it is almost certain that every possible single base change compatible with life exists in a living human on the planet Earth today. So we need to capture that and study it. And we've started doing this through variant aggregation at the Broad Institute. This was started in 2014 by Daniel MacArthur's group when he created the Exome Aggregation Consortium, or EXAC, which had 60,000 exomes, all mapped to Human Genome Build 37. This was the largest publicly available reference database of genomic information. He followed this with the Genome Aggregation Database, or gnomAD, which was released in 2016 with 125,000 exomes and added on top of that 15,000 genomes, also on Genome Build 37. More recently, the Nomad version 3 has been released. It has 71,000 genomes. There is overlap with many of the samples in the prior V2 version of Nomad, but these were actually natively mapped to Genome Build 38, and this was released in 2019 and is the largest reference database of coding and non-coding variation. You can see that these resources have been incredibly utilized by the whole entire community, and every diagnostic report that I get back today has a comment on the variant allele frequency or the presence of the variant in gnomAD. This is a graph of over time, starting from the first release of EXAC when it made its splash at ASHG, to seeing how this resource has been the use of this resource has been adopted in the genetics community with over 100,000 weekly page views, with a total of over 20 million page views over the life of the database from 184 countries. And we estimate, although we don't have specific data, that has aided the diagnosis of over 200,000 patients with rare disease. So who is in the gnomAD database? I told you there's exome and genome data. This data was not sequenced as part of gnomAD, but was aggregated from pre-existing case control studies of complex adult onset disease, such as type 2 diabetes, heart attack, migraine, bipolar disease. We have depleted it as much as possible of people known to have severe pediatric disease and their first-degree relatives. So we would never take a cohort that had been recruited for rare disease or Mendelian disease and sequenced, or the parents of individuals in that cohort, and include them in the database. It's pretty evenly split between male and female, although it's 55% male, and the average age of the individuals in this data set is 54 years. You can see that the data sets have grown over time, with EXAC represented here through number of exomes. gnomAD is even larger, as I mentioned, and Nomad V3 has a larger number of genomes and better representation of African ancestry, noted in purple here, although you will very much note the European-centric bias that is present in the available sequencing data, which is something that needs to be addressed with improved studies to include in these data sets. Again, this is aggregation of data from existing studies. This is a list of most, probably not all, as there are constantly new data sets being added of the PIs who share data in Nomad, and you can check out our website for the most up-to-date list. The data, really importantly, is all brought into the Broad Institute. The variants are all mapped exactly the same way. The data is jointly called in one giant uniform, parallel processed, really clean variant call file, or VCF, using Hail, and with a lot of support from the genomics platform and the data sciences platform. And this is how we're able to make this resource and release it to the community. So this is the landing page of the Nomad website. Anyone can go and access it at nomad.broadinstitute.org. Again, I'm going to show you a number of features on Nomad v2, which is currently the largest data set on HG37, and we'll mention a few features on Nomad v3, which is on HG38 and only contains genomes for now, but there is a plan to expand this to exomes in the near-ish future. I'm going to cover a lot of material here. We've had some recent updates, so you're actually getting the most up-to-date version here. I do have other recorded talks that have a lot of overlap with this talk, in case you wanted to check those out on other talks on using the Nomad browser that might go into a little more detail than I have time for today. So one of the things is, as we've added more and more features to Nomad, it's really grown as a resource, but it gets harder for me to fit it all on slides when I'm demonstrating. So I'm going to switch over and show you live using the Nomad browser. So you can type in any gene you're interested in. I really like the gene NSD1, which is involved with a condition called Soto syndrome, which is an overgrowth and intellectual stability syndrome, but you can look up any gene. You can look it up by gene name or on ensemble ID. You see the gene name here. You can tell what build we're in. You can see the different information about the transcripts and some links out. We'll get to more of that later. This is the constraint table up here. I'll also be telling you more about that later. And as you scroll down, you can see this is kind of the heart of the page here. So we have the exons in black. And on top of this, we have this coverage map that has the X-fold coverage sequence coverage for the exome data in blue. And you'll see there's some peaks and valleys as it varies with the GC content. And of course, we don't go into the introns for the exome data. And the genome data in green, which is a lot of, about a little over half of is PCR-free genomes. And you can see this is just very nice, clean coverage across the whole region. We are showing this shortening the introns, but you can actually click region view and see the whole gene. So as we scroll down, we have ClinVar variants here. Very important point. These are all of the variants in ClinVar, but that does not mean these variants are all in the Nomad dataset. In fact, most of these are probably not in the Nomad dataset. I recommend that you click to expand all variants, and then you can see all the ClinVar variants here. They're colored by their pathogenicity, whether they're pathogenic, a variant of uncertain significance, or benign, or some of these don't have an assertion in this database. You can hover over any of them. Actually hover over a pathogenic one. You can see that this is a pathogenic variant. The P.dot nomenclature is there. It's a frame shift, and it's been submitted by one submitter and has 1 star. And you can click there. Oops, not doing a good job there. Click on the dot to open the page in ClinVar. Okay, so as we scroll down, this is a track of all of the Nomad variants with the height representing how common the variants are. As we go further down, we basically get to a list of all of the variants seen in this gene. There's a column for clinical significance. There's a column for flags if there's some concern about this variant. It's highlighting this as a multinucleotide variant. There is the allele count, the number of times this variant is seen in the call set. The allele number is the number of chromosomes genotyped at this site, which is 2 times the number of individuals, unless we're talking about the X or Y chromosome. The allele frequency is the allele count divided by the allele number. And we also note the number of homozygotes here. If you want, you can sort by any of these columns. If you want to look at flags or clinical significance, it's all sortable. Now I've actually, so if I go back to the default is it sorts by variant ID position. And I can also search for any variant I'm interested in. I can search for every variant that has a phenylalanine in it. I can search for a specific, position, so you can filter this table a number of ways. We're only showing past variants here, but you can click and show variants that fail QC filters also. If you're interested just in loss of function variants in the gene, you can click just loss of function only, and then you can add other variants in as you want it. So there's a lot of ability to choose what you want to look at. Okay, so then let me just pick a variant here. I'm going to pick this missense variant. On any of the variants you can click and you can get to the variant page. This will tell you how often the variant is seen in exomes and how often in genomes, remembering that, again, there's many more chromosomes and samples in the exome data set than the genomes. It'll tell you the allele frequencies, which you shouldn't compare too closely because the ancestry breakdown is different across the two data sets. We give some basic polyphen and SIFT in silico markers there, which are transcript specific. It gives you the information from ClinVar here again that was present on the previous page. And then this is really the heart of the variant page. I'm going to zoom in a little here. You can see that this variant is present 33 times. It's actually only found in East Asians. And if I open this up, I can see it's actually found often in Korean or other East Asians. And I will note that the Japanese population in Nomad is very small, so I don't think you can really comment on that. And so you can just see where this variant is found. For example, we don't have subpopulations for all of these, but for European, the Finnish European, for non-Finnish European, we have a few additional ancestries. You can see the age of the individuals with this variant, and if you scroll down, you can see some metrics. So it is defaulting to all here, but if I select variant carriers, you can see all the variants have a GQ that's around 99. It's very high. You can see the depth of the variant or the allele balance for the heterozygotes, and that all looks good. And then you can actually scroll down and you can see the read data for the variant. And so you can take a look at that and make sure the variant looks good. There's no other variants around that are concerning. You can zoom in or out a little bit, but we are showing you just a subsetted BAM. So you're not able to scroll through the data this way, but you can take a look at the region and make sure everything looks good. And so this is basically the Nomad website. So going back to my slides, so this is what I've walked you through now. And I talked about the variant page and the different ancestries and looking at the read data. So there are a few additional features that I've showed you Nomad v2 data just now. There are additional features in v3, and I will show you in the slides how to switch between data sets. So in Nomad v3, we have added some in silico predictors, Revel, CAT, and Primate AI that are at least for missense variants. CAT is for really any variant, and Splice AI, which is these are scores by Illumina, but Splice AI is to predict variants that could impact splicing. And so the 0 means no expected impact on splicing for this variant. In Nomad v3, we have the Nomad data. I'm showing you a different, much more common variant here. So I would have some good allele counts. You can see it's represented across a number of ancestries, most common in South Asian here at 5%, but still 1% in European and Amish and rarer in Ashkenazi Jewish. So variants, it's present across all populations. So I'm showing these tabs where there's 2 additional data sets just present in Nomad v3. And these are the Human Genome Diversity Project and the 1000 Genomes Project. And so you just click over on one of these tabs, you can see for the same variant, the data that is subsetted to this publicly available data set, HGDP. And if you click on any of these little triangles, you can expand that and learn more about some of the subpopulations that the data was collected from. And also, if you're interested on any of these variant pages, we also have the number of XX samples that have the variant and the number of XY samples, so males and females. Okay, so lots of information on in the Nomad data. One of the things I mentioned very briefly was the constraint table in the top right-hand corner of every gene page. And what is constraint? So we talk about conservation, which is how similar a position is or a region is across different species. constraint is the same kind of concept, but it's how similar is a region or a variant within the human population. And so the idea here is if this is just looking at missense variants, you can see this is a gene. These are the exons of a gene. So the introns have been removed, just the coding region of the gene. And we're graphing here the density of missense variants seen in Nomad in the general population. And so you can see there are some regions with a fair number of missense variants. There are other regions that really you don't see missense variation. And the fact that you don't see it likely means that it's not tolerated in the human population, or we would see it occurring by chance given the number of people we've sampled. So when we layer on what we know about the protein, we can see that there are actually some protein domains here. So a PhD domain, another domain that I don't know the name of from the schematic, and you can see that the amount of variation is depleted in these regions with these domains. So there's a tie between depletion for variation and seeing functionality if you do see variation there. So Constraint is based on a mutational model that was developed by Caitlin Samoha and Konrad Karczewski, along with Daniel MacArthur and Mark Daly. And the basic idea here is that they calculated the number of expected single nucleotide variants. So this is just SNVs, not including indels in this model. Single nucleotide variants that would cause a synonymous change, a missense change, or a predicted loss of function, which includes nonsense and essential splice sites. And they asked how many of these do we expect for each gene, given what we know about mutational rates. And so this is actually for NSD1, the gene I showed you before, and we expect to see 525 synonymous variants. When we look in Nomad, we expect to see 525 synonymous variants in a population of 140,000 people like we have in Nomad. And when we look in Nomad, we see 523. For missense variants, we expect to see 1,427, and we see 1,065, so a little bit lower. And for loss of function, we expect 110, 111, and we only see 5. So that's very significantly depleted from expectation there. And so how do we assess this depletion from expectation? The first thing that they did is they generated z-scores, so standard deviations from the mean of expectation there. And you can see for synonymous, you see basically observed over expected, just about what you expect. This is the 95% confidence interval on that. For missense variation, we do have an elevated z-score of 3.4, which we highlight yellow, meaning we think that's significant. We turn it yellow at 3.1 or 3.09. And you see about 75% of expected missense variance in this gene. For our predicted loss of function, we have a PLI of 1 and a LUF of 0.1. And these are two different metrics that we use to assess significantly depleted for loss of function variation or loss of function constrained genes. We have these two different metrics that are different than the z-score because the number of expected loss of function variants is lower. And when you're dealing with these lower numbers, you can start to see the power to detect depletion from expectation is significantly correlated with the gene length, whereas for missense variants, the numbers tend to be higher, and so there tends to be less sensitivity to the gene length. So these two metrics, why do we have two and what are they? So the first PLI is the initial metric developed by Caitlin. This is the probability of loss of function intolerance. It's a dichotomous metric, meaning it's a lumping metric. And it's good for if you want to draw a line and say, these genes above my line, we typically use 0.9, are loss of function constrained. On the other hand, this is biology. Some genes are going to be very loss of function constrained, and some will be a little bit, some will be not at all. The actual picture is much more of a continuation or spectrum. And so that's what Konrad developed LUFT for, which is loss of function observed over expected upper bound fraction, which just really means it's the upper 95% confidence interval of the observed over expected. It's a continuous metric with here lower scores or more loss of function constrained. If you're going to use this metric, closer to 0, again, more loss of function constrained, but we don't recommend that you draw lines. If you want to draw a line and say everything below or above this line, then you might want to use PLI. But as we look at the spectrum of loss of function intolerance, and we look at take genes that we know whether they're haploinsufficient, recessive, or tolerate loss of function, this is what we see. So again, loss of function depleted genes will be over here on the left, close to 0, and loss of function tolerant genes will be closer to 100. If we take known haploinsufficient genes where loss of one copy of the gene results in disease, we can see that those genes in the general population as a group are depleted for loss of function variation. If we take autosomal recessive genes where having loss of one copy is tolerated, you're a carrier, but if you lose both copies of the gene, then you have a condition. Those genes are seen across the spectrum, but have an average depletion centered around observed over expected of 60%. And if you take olfactory genes, of which we have hundreds, and there's redundancy, and you can lose different olfactory genes, there's not constraint on them, you see that these are clustered over very elevated Loof scores, or Loof scores on the higher end of the range. So a few caveats to constraint. It is primarily for dominant disease genes. We don't expect to see strong constraint signals or depletion of loss of function variation for recessive disease genes, but we do sometimes see things in the middle for Loof. For PLI, recessive disease genes tend to have a PLI closer to 0. So if you are wanting to see recessive, you might want to be thinking more about Loof using Loof as your metric. Importantly, it's not necessarily a sign of a strong phenotype. So you can see evidence of constraint from mild degrees of negative selection. Additionally, it can be a phenotype that is subtle from the perspective of seeing someone. For example, infertility has a large evolutionary detrimental effect in that you don't tend to pass on variants that result in infertility. But on the other hand, those individuals could absolutely be in Nomad, healthy people walking around in the population who may not even know they're infertile. The next point is that selection occurs through reproduction. Again, we're looking at variation over in the current population that's been passed on over the course of years. And so deleterious effects from the variant must be before or during reproductive years. And I just want to use BRCA1 as the example. So we know loss of function of BRCA1 results in an increased risk of breast and ovarian cancer. However, much of this cancer risk is after reproductive years. And so that's why we don't see a significant depletion of loss of function variation in this gene when we look in Nomad. Well, initially, I kind of introduced the idea of not just looking at the level of the whole gene, but the whole gene is how we think of loss of function constraint. We also have the metric for the whole gene for missense constraint. But we can also think of missense constraint as parts of genes, like overdomains, or they're biologically important parts of genes that have depletion of missense variation that we should be thinking about. And so what we're looking at here is regional missense constraint, also developed by Caitlin Samoha. And you can see this portion of the gene has only about 4% of expected loss of function variation. And so this is a very constrained region next to a less, also constrained. Whereas over here, you see 117% of expected variation. So if I saw a loss function variant right in this region, if I saw a missense variant in this missense depleted region here, that would definitely catch my attention more than a missense variant over in this region where we see a lot of missense variants in the general population. So this data is actually currently only available for EXAC, our original data set, but Catherine Chow and the group is working on updating this and getting this available on the Nomad data. If you want to change data sets, you just click here in the Nomad browser on the page view or the variant page, and you'll get a whole list of different data sets, so EXAC, Nomad, or V2 or V3, and so you can just flip between those data sets. You can only flip between V2 and V3 on the gene page. On the variant page, you can only look within a data set because of the different genome builds. I wanted to point out one other external site that's very helpful. So we've linked to a number of external sites. If you, this little menu is on the gene page right under the gene name for every gene. And so if you click the and more, it opens up this additional page. And one of the things I recommend checking out is Decipher. So you click Decipher, it takes you to another page. You might need to just click the protein view on that page, and then you can take a look and you can see. So this is the exons here for this NSD1 gene I've been showing you. This is the same regional missense constraint data that I just showed you on the exact browser. You can see the density of missense variation over that. And then this is showing you all the domains here. So you can see this region that is depleted for variation does have a number of protein domains. And you can see in red all the pathogenic and likely pathogenic variants. The open triangles are the variants of uncertain significance. And then, the open ones, yeah, variants of uncertain significance. The yellow ones are missense variants, and the red ones are loss of function variants. So a lot of data on this site also, similar to the data on the Nomad browser, but it's another way of viewing a lot of this. Okay, there's also expression data from the genotype tissue expression project that's available to view in the Nomad browser. So I showed you initially on the gene page that you have the exons here. If you were to click show transcripts, it would open up the GTEX data here. It shows you the mean expression of different isoforms. Sorry, it shows you the expression of different isoforms here. So this here is showing high expression. These are all lowly expressed. And this is for the second transcript, is the most highly expressed transcript. If I was interested in a particular tissue that I wanted to see the expression, I could click this mean bar here and I could select which tissue I wanted to see. Select that, it would change the graph. And then I just wanted to briefly, you can go out and you can see the GTEX data is all available in the GTEX portal. I will say that there's additional data here, both gene expression, transcript expression, and eQTL data located on the site. This is a research study where there are 53 tissues collected from 714 people who donated their body to this project after death, and on autopsy, all of these samples were taken and RNA sequencing along with exome and genome data was generated. These are generally adults, these are all adults, and generally of kind of 50 to 60 age range. And so it's not a great data set for developmental disorders, but it's still one of the largest and most uniformly processed data sets. You can click on any gene in the GTEX data and you can see the tissues it's highly expressed in. I'll say I actually often use this if I, for example, wanted to do a follow-up study to look at expression or splicing in a tissue that I'm able to easily access. These are cultured fibroblasts here, these are LCLs here, and this is whole blood. So I can often use it to figure out what follow-up studies I might be able to do. Okay. We also work by Burrell Cummings has developed something called a PEXT score, or a proportion expressed transcript score. And this basically looks at all the transcript information and the GTEX data, and it asks what part of which exons or which part of the exons are actually present in the RNA-seq transcript data. And so you can see, for example, here, this is kind of these two exons or one big exon, which one is actually the normal part that's transcribed. And when we look at PEXT, we can see these two little exons are transcribed. This exon here that's only present in one transcript, actually not transcribed most of the time, whereas these exons are transcribed, and again, not transcribed. And so you can see that there are loss of function variants in ClinVar, again, variants that are not in Nomad throughout the gene. But when we look at loss of function variants that are present in Nomad, they are mainly falling in this part that's not transcribed here, this exon that's not transcribed here, and also in the last exon. I will say there's this one here that you can't obviously explain just by looking at this view, but when you look into this, it's a splice site variant, and there's actually a nearby in-frame rescue splice site. So I think that explains why this variant is here. And so this is often the case that we will see loss of function variants in genes that we know have severe phenotypes when there is loss of function of the gene. And when we look at these variants in Nomad, we can often explain a good proportion of them and why we see them and how they're not actually resulting in loss of function. So I mentioned that you can just click on the Nomad little menu here, and you can change the data set to a subset. So for example, if you're studying cancer and you want to know if this variant in this one that is seen in one African individual if that variant comes from an individual who's in a cancer cohort or not. You can click the non-cancer, and that will exclude any samples that are in the cancer cohort. And you can see this variant is not present in the non-cancer subset because the variant is only present in the cancer subset. There aren't cancer samples, so these are not tumors that are sequenced and included in Nomad. They are blood samples from people who were recruited from a study of cancer. So they're germline blood samples for non-blood cancers, but generally, just sometimes knowing what subset something is in can be helpful depending on the question you're asking. But for most uses, we just recommend using the full Nomad data set. There's also a region view. If you click here on any gene page, it will show you all of the variants for all the intronic regions also, in case you need that. There's also a Nomad structural variant data set. So if you click over here on Nomad SV, it's going to take you to another page. Before I get to that, I'm just showing you the paper briefly. So this is a data set generated by a number of people, but led by Mike Tolkowski with the work led by Ryan Collins and Harrison Brand. The call set is done on about 11,000 genome samples. They use multiple algorithms along with integration step and a lot of QC to generate a high quality SV reference set. And there are over 400,000 unique structural variants with frequency data. And I'll say I found this very useful clinically. Often when I do see parents who are carrier for a deletion, I can often find that deletion is present at low frequency in the general population. Just consistent with why I could see it in my carrier parents. So I'm just showing you an example here of the RBM8A gene. We know that this is associated, deletions of this gene, a deletion encompassing this gene and additional genes are associated with thrombocytopenia absent radius or TAR syndrome. It's a recessive condition where generally affected individuals have one deletion in trans to a single nucleotide variant. And so I was curious how often we see this in the general population. I actually initially was trying to see how the estimates from the literature compared to the estimates we see in Nomad, but I actually could not find a good estimate in the literature for how common this is in the carrier state in the general population. And again, just highlighting how we just don't have a lot of good access to this data, and these resources are really powerful. So you can see here that in blue are copy gains and red are loss of function. So I'm seeing here a loss of function variant. It's a deletion, it's 240 kb. If I click here on that line, it actually scoots me out. And then I zoomed out a little bit. So you can see this is the deletion. It deletes a number of genes, including the RBM8A gene. This is the previously reported deletion for TAR syndrome. If I click here on the gene, on the variant, it will take me to the variant page for the structural variants. And you can see it's seen 3 times in this population, 2 Europeans and 1 African. And so I could also check if it was in controls or non-neuro samples. But I don't really see a need to do that because I wouldn't expect any phenotype in carriers of this variant. But overall, this is seen very rarely, but is seen in the general population. We have some new data sets that we have been recently added. So last October, we added the mitochondrial variants to Nomad V3. They're not in V2, but they're part of V3 because they're only part of the genome call set. And so the idea here is that mitochondria are evolutionarily derived from bacteria, and they have their own DNA in them. And so it's this circular DNA. It has 37 genes. It's about 16 kb. And each mitochondria has multiple copies of this circular DNA. And each cell in your body has thousands or hundreds of thousands, I don't even know how many, mitochondria. And so you have many, many, many copies of this mitochondrial DNA. And when we do genome sequencing, I think we get about 5,000x coverage on average of the mitochondrial genome. A number of diseases have been linked to variants in mitochondrial DNA. And so it's important to have a good reference for this data too. Oops, I forgot to, maybe I get to that later. So sorry, okay. So this is, if you type in any gene that's on the mitochondrial DNA, so I'm just typing in MTND5 here, which I randomly selected as a mitochondrial gene to show you. You can see the coverage of the mitochondrial genome, and on average, it's almost 3,000 fold across the data. We've just yesterday, or I guess last week by the time you see this, added the ClinVar variants. That was done by Nick Watts, who's is the one who really leads the development of and getting new data into the Nomad browser. And then you can see all of the Nomad variants in the mitochondrial genome here. We do have some additional information, and in addition to clinical significance, we have flags for common low heteroplasmy, the allele numbers, the homoplasmic allele count, the heteroplasmic allele count, and then the maximum observed heteroplasmy level. So for anything that is seen in homoplasmic, that'll be pretty high. Okay, so same kind of idea here. You can click on any of these variants. So as I click on one variant, I can now see here again all the data I just told you about from the table, the ClinVar information. I can see the heteroplasmy level for this variant that is seen in in a handful of individuals, 2 homoplasmic and 2 heteroplasmic. And then I can come down to this table here and I can see the mitochondrial haplogroup and see where these are seen. So they are seen homoplasmic in an individual with M, an individual with the U haplogroup, and they're heteroplasmic in an individual with the H and an individual with the L2 haplogroup. So we hope this data will be very helpful. And again, this is work that was done by Vamsi Mutha, along with Sarah Calvo, Kristen Laricha, Monko Lek, Nicole Lake, and Daniel MacArthur. Loss of function curation. So it's important to remember that when we say something is a loss of function variant, it's really a predicted loss of function variant, and it's predicted by a computer. And so we often can look at variants and see things by human review that make us think that maybe the variant isn't really resulting in loss of function. And so we have curated thousands of variants, but there are hundreds of thousands of variants, loss function variants in the data set. And so we will never get to all of them, but we have done some that we think are interesting, and this is a continual endeavor led and largely done by Moriel Singer-Burke, along with a number of other individuals who have curated this data that you can read more about here. So they have commented on whether they think the variant that is called loss of function is likely to result in loss of function, or if there's some reason why it is not. For example, this variant is found in a homopolymer and there are some mapping issues. So we have curated all the homozygous loss of function variants, along with some handful of heterozygous recessive disease genes, and some haploinsufficient genes that are not quite added to the browser yet, but will be coming in the future. So if there is a loss of function curation, this column will be present on the gene page, but for the majority of genes, we do not have this data. So genetics is a really important lever to understand biology, and by doing large-scale sequencing and reference data like this, we can do forward genetic studies where we study people with disease and we compare them to existing data sets, or we can take large-scale genomic studies like the Nomad and find interesting variants, and hopefully one day be able to follow those up with a reverse genetic study to better understand the phenotypes from those variants, for example, with loss function variants in the general population. Nomad, for the most part, is not recontactable, so this is not something we're able to do, but as we have biobanks like UK Biobank and all of us studies, hopefully there'll be a more increased ability to link these large databases with medical records and the ability to recontact for deeper phenotyping. So in the last minute, I just want to comment on harnessing the power of allele frequency. To make sense of one person's exome or genome, we need tens of thousands of exomes or genomes. And this is highlighted here by if we so we all have in our exome, this is exome data here, if we all have about 20,000 variants in the VCF, if we were to ask what are the very rare variants, less than 0.1% allele frequency, if we used a small data set of 6,000 people that only had European and African ancestry individuals in there, we would actually be left with about 600 to almost 1,000 rare variants, very rare variants to follow up on. And it would work better in the ancestries represented in this data set and less well in underrepresented ancestries, highlighting the importance of ancestral diversity in these data sets. When we moved to EXAC with 60,000 individuals and improved ancestral diversity, at least some representation of each of these groups, we were left with only about 150 variants that were very rare per individual. And these databases have become essential in the the standards and guidelines for interpretation of sequence variants from the American College of Medical Genetics, which I think you'll be hearing a lot more about throughout this workshop. And I just wanted to highlight, we think about using Nomad data when a variant is too commonly seen in the population to be consistent with the disease prevalence or mechanism, or when a variant is absent from the general population or possibly at very low allele frequency for recessive variants. So just the allele frequency alone is criteria that we consider in interpreting every variant. And then here, these mutational hotspots, missense variants with low rate of benign variation, these also give us some sense of constrained regions of the genome. And so we can start to think about constraint and what we can learn from human population in using these criteria also. So I hope I've convinced you that reference population databases are public resources that are critically important to evaluate variant rarity, which is a necessary but definitely not sufficient step for pathogenicity for rare disease. These datasets provide multiple tools, constraint, PECs, to help prioritize variants to understand human biology. And the power of reference population datasets will increase as they grow in size and, really importantly, in diversity. Nomad V4, we hope to have possibly later this year or in early 2022, which will have over 300,000 exomes and will be on genome build 38 and continue to incorporate the genomes that we have today, and we'll be growing that over time also. So I've shown you how you can access this data through the website. You can also download the entire data set or incorporate it into your pipelines. We've made it available along with partnerships from Google Cloud, Amazon, AWS, and Azure to host these data sets in open, accessible places. And so you can go to the Nomad page and just go to the downloads subpage, and you can find information about how to download all of the data, from the variant data to the constraint data, to the structural variants, and many of the things I've talked about today. I just want to point out there's a common misconception that the genome data is the non-coding data and the exomes is the coding. It is not. The exome data is literally the 125,000 exomes. The genome data is the 15,000 genomes. And so if you just got the genome data, you would be missing 125,000 exomes worth of data. So check out whatever you need for your analysis. And I've covered a lot of things here. I know I've gone a little faster than I had hoped to. Nomad has a blog on the news tab of the Nomad page. You can read in very great detail about all of our recent releases and more about these features, so you can catch up on anything that you missed here. And this is a huge team effort supported by a lot of people across the Broad, along with support from the Broad, along with the NIH, to make this resource that is available to the entire world. And we're very proud to be part of it. So check it out on the website. If you have questions, you're welcome to reach out to me, or you can contact the Nomad team directly at nomad@broadinstitute.org. And you can read about the research that has come out of the Nomad data set in this paper collection here. So thank you so much for your time today, and I wish you all the best in the rest of the workshop, and I'm happy to take any questions.